In the previous sections, we discussed overall microbial immunity and bacterial structure and antigenicity. Now let's dive deeper and have a closer look at the individual aspects of interest of various bacterial specie. First up is Streptococcus. Well, streptococci are divided into 3 groups by the type of hemolysis on blood agar. Those that show an incomplete green hemolysis are called alpha hemolytic. Those having clear, complete lyses of red cells are called beta hemolytic. And if they show no hemolysis at all, are classified as gamma hemolytic. While there's another system of classification, serologic grouping is based on antigenic differences in cell wall carbohydrates, group CT-V, in cell wall pili-associated protein, and in the polysaccharide capsule in group B streptococci. Now, moving on to the virulence factors of group A streptococci, they include M protein and lipoteichoic acid for attachment, a hyaluronic acid capsule that inhibits phagocytosis, other extracellular products, exotoxins such as pyrogenic, erythrogenic, toxin, which causes the rash of scarlet fever, and invasins like streptokinase, streptodornase, also known as DNASB, and streptolysins. Some strains of streptococci are nephrogenic. That means they can cause nephritis, while the non-group A strains have no defined virulence factors. Now, keeping all this in mind, how do you think your body is going to defend you against streptococcus? Well, antibody to M protein will give type-specific immunity to group A streptococci, while antibody to erythrogenic toxin prevents the rash of scarlet fever. Immune mechanisms are also important in the pathogenesis of acute rheumatic fever, and maternal IgG protects the neonate against group B streptococci. Next bacteria we are going to discuss is Staphylococcus. Talking specifically about S. aureus, it expresses many potential virulence factors, including surface proteins that promote colonization of host tissues, factors that probably inhibit phagocytosis, like capsule and immunoglobulin binding protein A, and toxins that damage host tissues and cause disease symptoms, while the S. epidermides readily colonizes implanted devices. Now, in host defense, phagocytosis is the major mechanism for combating staphylococcal infection. Antibodies are produced, which neutralize toxins and promote opsonization. However, the clever Staphylococcus has antiphagocytic factors, including the fibrin-forming enzyme coagulase and protein A, which binds to the Fc portion of IgG, thus blocking opsonization. Not only this, but there are numerous other toxins that make staphylococci highly destructive, abscess-forming organisms. Well, large-scale use of antibiotics has caused the emergence of bacterial strains resistant to many antibiotics, like methicillin-resistant Staphylococcus aureus, or MRSA, which are now proving a serious threat, particularly as hospital-acquired infections. Next, I am going to talk about pneumococcus, or Streptococcus pneumoniae, and meningococcus together. Well, they are typed by the polysaccharides of their capsules, and are especially virulent in the tropics, where vaccines made from capsular polysaccharides are proving highly effective in preventing epidemics. Also, they are more common in patients with deficient antibody responses. The chemical coupling of the capsular polysaccharides to a protein, such as diphtheria toxoid, converts these antigens from T-cell independent to T-cell dependent, thus greatly increasing memory and potency. Such conjugate vaccines have proven highly effective at preventing childhood meningitis and Haemophilus infection. Now, the bacteria responsible for gonorrhea. Gonococcus. Gonococci, adhere to columnar epithelial cells, penetrate them, and multiply on the basement membrane. And their adherence is facilitated through pili and opoproteins. The lipopolysaccharide of gonococci stimulates the production of tumor necrosis factor, which causes the actual cell damage. Ig may block attachment to mucous surfaces, but the bacteria secrete a protease that destroys the IgA. Thus, the infection is seldom eliminated, leading to a carrier state. Gonococci and meningococci are the only bacteria definitely shown to be disposed of by complement-mediated lyses. Well, gonococci may also disseminate via the bloodstream, and these strains that cause disseminated infections are usually resistant to serum and complement. Moving on to the next bacteria that are Mycobacterium tuberculosis that causes tuberculosis, and Mycobacterium leprae that causes leprosy, also known as Hansen's disease. These mycobacteria have very tough cell walls, rich in lipids, which resist intracellular killing. Moreover, they can also inhibit phagosome-lysosome fusion. Well, chronic cell-mediated immunity results in the formation of granuloma, tissue destruction, and scarring. Now, the spectrum of leprosy ranges from lepromatous to tuberculoid, where lepromatous being disseminated, multibacillary, with loss of specific cell-mediated immunity, and tuberculoid being localized paucibacillary with strong cell-mediated immunity. Next up is Escherichia coli, or E. coli, which is now perhaps the best-known bacterial species in the world, and that's because of its ubiquitous use as a tool in all molecular biology laboratories. However, the species is made up of an enormous number of different strains. Virulent strains differ from non-virulent E. coli only in possessing genetic elements for virulence factors. Strains producing enterotoxins are enterotoxigenic E. coli, or ETEC. Here, gastric acid and intestinal transit time are important defenses against E. coli. Specific intestinal immunoglobulin A, or IgA, develops and appears to be protective. Salmonella, one example being Salmonella typhi, that infects the intestine, but can survive and spread to other parts of the body within macrophages. Recovery after infections may lead to a carrier state. So, to protect us, our body chooses both specific and non-specific host defenses to be active against Salmonella. The specific defenses consist of mucosal and systemic antibodies and genetic resistance to invasion, while non-specific defenses consist of gastric acidity, intestinal mucus, intestinal motility, peristalsis, lactoferrin, and lysozyme. Next we have tetanus causing Clostridium tetani. Well, tetanus owes its severity to the rapid action of its exotoxin on the CNS. Here, antibody or antitoxin is highly effective at blocking toxin action, an example where neither complement nor phagocytic cells are needed. Moving further to diphtheria, caused by Corynebacterium diphtheriae. Well, it also secretes powerful neurotoxins, but death can be due to local tissue damage in the larynx, or false membrane. Talking about host defense, it involves an antibody response to diphtheria toxin following clinical disease, or to diphtheria toxoid, that is, formaldehyde inactivated toxin, following immunization. Now, lastly, I would like to mention another sexually transmitted disease, syphilis, caused by Treponema pallidum. Well, it is an example of bacteria surviving all forms of immune attack without sheltering inside cells. Treponemes are highly invasive pathogens which often disseminate relatively soon after inoculation. Although treponemes lack classical lipopolysaccharide, endotoxin, they possess abundant lipoproteins which induce inflammatory processes. Now, the defense mechanisms our body choose against treponema are both cellular and humoral. However, clearance of treponemes from local sites appears to be due to phagocytosis by macrophages. Borrelia. Another spirochete has the property, found also with some viruses and protozoa, of varying its surface antigens to confuse the host's antibody-forming system. As a result, waves of infection are seen relapsing fever. And Brucella may do the same. So now you must be thinking how clever these bacteria are. That was all about immunity to bacteria. I hope you have enjoyed the video. For more such videos, keep watching sqadia.com. And don't forget to give your feedback.